Hello, good evening and good day, everyone. Welcome back to my IVF Answers and the European Fertility Society. We are back once again with an amazing and very difficult topic, I would say, as well. Still a new topic as well. So I'm really happy and excited to have our special guest with us today. Hello, great to see you. Professor Shoukhrat Mitalipov is with us. Hello, Professor Mitalipov. It's great to have you. Thank you so much for agreeing and joining us today. How is your day going? Are you ready to educate us on our topic today?
Yes, I'm ready. It's still morning here, so I'm fresh.
Definitely glad to
Ready
hear
to
that,
go.
of course. It's true. It's 11 a.m. for Professor Mitalipov. It's a little bit later for us, but we are here to discuss a very important topic, still new topic, as I've mentioned. And Professor Mitalipov will talk about spindle transfer, but all other mitochondrial donation replacement therapies techniques, just to give us an overview analysis of his research, because this is his main research. This is something that he's the best person to discuss for sure as well, and as it is focused on MRT. And just let me remind that Professor Mitalipov is director of Center for Embryonic and Gene Therapy at Oregon Health and Science University. So we are definitely happy to have you here. Before we start, as always, let me remind you that we will start with our presentation on the topic. And afterwards, your favorite part, you will have a chance to ask your questions. So remember, it's all anonymous, so you can type all the questions in the chat. Professor Mitalipov will be happy to answer them for you right after his presentation, I'm sure. I'm really looking forward to this session, and I'm sure you will have lots of various questions, so don't hesitate. It's also being recorded and will be available on our YouTube channel, as well as my IVF Answers website. And with that, I think we can go ahead and start our presentation. Professor Mitalipov, are you ready to begin?
Yes, I am. Thank you, Caroline, for a nice introduction. And
Thank you so
hi,
much.
hello, everybody. And as let me pick the as Caroline pointed, I'll be talking about the mitochondrial replacement techniques, or sometimes we call them therapies, and in short, it's MRT. Primarily, I'll be talking about the spindle transfer. That's kind of one of the most advanced form of MRT, but I'll also tell a little bit about some other kind of alternative techniques, which is also, it's called MRT. So I do primarily research here at the Oregon Health and Science University, and uh, we of course do research on eggs and sperm and embryos, with the idea of developing new therapies, cell therapies and gene therapies that would be introduced into IVF. So how advanced? It's advanced. So as you know, IVF, or in vitro fertilization, has been introduced many years ago, and it's started as a simple kind of mixing egg and sperm that we retrieve from patients in a petri dish. And fertilization occurs, normally, just like it would be in a uterus, and that's usually what started IVF. But since then, of course, the IVF becomes more complex. So it has actually a number of kind of techniques or technologies that today we all call IVF. So among those techniques, of course, as I said, there are core techniques, and one of them, of course, is just a fertilization, where you could get the egg from a patient and sperm from a from a partner, and you just mix it up, and voila, it actually works. But of course, now we can actually fertilize it by helping with injection of the sperm. So this is the picture, which we call intracytoplasmic sperm injection, or sometimes we just call it ICSI. And this is actually prescribed when there is a low sperm count or low motility. and that means regular fertilization would not occur if you mix it up. So we kind of pick a sperm and then use this pipette to basically directly inject inside of the egg and then let this sperm, into cytoplasm. So that's another form of fertilization. So sometimes we call it ICSI versus IVF. It means just mixing up. So the other kind of core techniques is, as I mentioned, it's embryo culture. We can now culture this embryo up to 5-6 days. So this is actually very important. So we can pre-screen for embryos that would arrest early, like day 2 or 3, many of them arrest. So we can grow to day 5. And of course, the early days, we could only say retrieve maybe that naturally cycling follicle, and that only get one egg. And of course, the timing would be very difficult. We have to wait. But now doctors do so-called controlled ovarian stimulation. So basically, they can plan for a specific day when the egg would be ready. And it's not only one egg. We actually can recruit multiple follicles at once. And that's why instead of single egg, we could get 10, 15. But that, of course, if patients are younger, with older patients, unfortunately, we still get very few eggs. So we could, of course, cryopreserve either eggs, or sperm, or embryos. And then other techniques that we use today is use of donor gametes, especially with advanced maternal age. Eggs from the patients have a poor quality and they can never produce viable pregnancy. So we often offer them, use younger donor gametes, donor eggs, which is now becoming, alternative, I guess, the only option for women of advanced age. And of course, there's a surrogacy and so on. But these are core services that we provide, which means they actually work. There is no question about it. But IVF is actually a little bit more than that. Those core, these techniques. There are so-called add-ons, and there are so many of them actually today. Unfortunately, those haven't been really proven to be effective or safe even, and they actually optional. But many clinics offer, and patients sometimes sign up, but they should know that this is actually never been proven to be working. So one of these, I guess, well-known add-ons, especially I say this is like pre-implantation genetic testing for aneuploidy, so-called PGTA. And actually here listed a few the embryology-based add-ons that never been proven neither effective or safe. But if you go to, say, HFEA, UK government's agency that regulates IVF in UK, they actually have a list of more of others. So among those, unfortunately, I have to say the MRT is another one. So it's still unproven, even in terms of safety. So this is what we're going to discuss today, and of course, whether it's actually really has any effect on certain types of infertility or genetic disease. That's a topic of my presentation. So why we actually use some of the, for example, MRT is primarily to improve egg quality of women advanced maternal age. As you know, as woman gets older, her ability to produce viable pregnancy, in this case, live birth, declines. And this is in vitro fertilization data. It's not natural, but natural probably follows the same rate. So as you could see, by age 35, the ability to produce live pregnancy with IVF with their own eggs declines drastically, and it goes to almost to zero by mid 40s. And unfortunately, many patients actually come to the IVF at this age. So this is infertility that actually caused by age. So it's a kind of aging disease. However, if the same patients would take, as I said, donor egg, and of course, donor eggs are usually young, proven donors, the live birth rates for the same woman is actually doesn't change. As you could see, they could have a pregnancy, a normal pregnancy, up to 50s even. So having that trend, of course, the clinics now offer egg donation to patients at this stage. But of course, the issue is that there is no genetic link from mom to offspring, and that's why patients unfortunately go through many cycles with their own eggs trying to get pregnant before they actually kind of forced to take donor eggs. So as we know, because of this data, the whole issue with low pregnancy rate is egg quality. And of course, there is a number too, as I mentioned. At younger age, we can actually retrieve 10, 15, 20 eggs, but the number of follicles that we can grow, even with hormones, declines, and at later stages, we can get just a few or just a one egg, and of course that's another contribution to the low pregnancy rate. But primarily, it's the egg quality. So we in my lab, we've been trying to understand what's actually causing this decline in egg quality. And the simple question is it because of sperm's egg, or the sperms is no problem, so it's an egg issue. So it's an egg's nucleus, or it's an egg cytoplasm. As you know, eggs are large cells in our body. They carry huge cytoplasm, and of course as nucleus as any other cell. So we can actually separate nucleus and cytoplasm, and this is how we do it. We have a certain procedures, which is actually today's we use for spindle transfer. So in this case, we separate spindle with mature egg and then cytoplasm. And now you could ask, what is it causing it? And research actually shows lots of issues are due to cytoplasmic problems or deficiency. So the cytoplasm has many functions, and actually the cytoplasm is probably the part of the egg that makes life come true. So we actually call it totipotency, or the totipotency inducing activity. So that means DNA, the nucleus of the egg and sperm, they actually they don't come as embryos. So, but they become embryo life as new life because of the cytoplasm of the egg maintains these core reprogramming factors, which we call totipotency, and they actually accumulated there during egg maturation and growth. And by the time it's mature, they have to be all set to go. But they will wait fertilization, and then after fertilization, they will be released or activated, and they will start working on the DNA of the egg and sperm to make it embryo, which we call totipotency. And so that's actually one of the main function of the egg cytoplasm. As you could see, that's what gives the life to the embryo. So it's that important. Unfortunately, it rarely discussed, and most people who look into egg cytoplasm, they only say about mitochondrial mtDNA. Yes, that's part of the cytoplasm. It's organelles, of course, it has its own DNA, but they are probably not the main contribution to the aging, even though we cannot exclude. So there is other factors that egg cytoplasm that defines what the embryo is. It's actually meiotic and mitotic factors, and they induce haploidy and then support pre-implantation mitotic divisions, meaning they're responsible for proper chromosome segregation. And if anything goes wrong during egg maturation, and these factors are not present or missing, now you get aneuploidy, right? Now you know where the aneuploidy comes from. Of course, there's other factors that support embryo development. As you know, the after fertilization, embryo will develop, especially first 5 days after the blastocyst stage, purely by support of the egg cytoplasm. So if you know that's the energy, but also lots of other proteins and mRNA, and it's also important. And if embryo or egg didn't accumulate all these supporting factors, you have embryo arrest early, right? So now you could see how important egg cytoplasm is. Whether the DNA, it's a nucleus, is just a template, you could actually replace it with any DNA as long as it has all full set of genes required for development. And that's how, sometimes we, in my lab, we replace that egg nucleus with skin nucleus, say, or any other, and that's actually not egg, it's not an embryo, but that becomes embryo. We actually call it cloning, but this tells you that egg cytoplasm, that's all you need to have a life, of course, and DNA template. Anyway, so giving that so much is depends on embryo from the egg cytoplasm. Of course, the question is whether we can replace it. Say if there is a poor quality egg, particularly for patients from advanced maternal age, well, can we say retain nucleus, but cytoplasm, which is bad part, we can just throw it away and we will take it, the cytoplasm from donor egg. Nowadays, we take a whole donor egg with its nucleus, and that's why the baby is not genetically related to mom. But in this case, the nucleus, which has a DNA from mom, would be replaced. So this is now the question is, can we now improve egg quality for these patients? So that's why we'll be doing, some of the procedures on the replacement, which is I'm gonna tell in my presentation. So replacing cytoplasm can be done in kind of different stages during egg development. One way is early immature egg. It's actually still in uterus, in ovary, and then but we still can retrieve it. So this egg is not mature because didn't become haploid, so it still has a visible nucleus. It's called the germinal vesicle stage. So you can transfer at this stage the nucleus, say, from patient into cytoplasm, but cytoplasm has to come from the same stage GV from a donor. So that's one of the principles of nuclear transfer. It has to go to the same stage, exactly the same stage of the donor egg. So that's one way to do it. And then, of course, this is when egg is matured. This is fully matured, ready to be fertilized. We call it metaphase 2, arrested oocyte. In this case, unfortunately, as you can see, there is no nucleus there visible. Hey, see, there is a big nucleus there. That's why it's so attractive to do, because everybody can see it. But at this stage, it's invisible because there is no really nucleus there per se. It's a chromosome scattered in this spindle, and that's why there is no nuclear membrane like in here, and that's why we cannot see it. So at this stage, transfer also can be done, which I developed, many years ago, almost 15 years ago. It's called spindle transfer, or maternal spindle transfer code. So there is another form of nuclear transfer that, this polar body. Could see there is a tiny cell. It's actually second egg. And it's actually contains DNA just like it would be inside, and that DNA is another, half of the haploid set, which is very attractive to transfer. In this case, it's just a nucleus of the second nucleus of the egg. So I'll be telling a little bit about that procedure, even though this hasn't been done yet. The other form of nuclear transfer is done after fertilization. So this egg would be fertilized with sperm, and then next day it becomes now embryo, but it's still kind of single cell embryo. We call it zygote. And in this case, you could see two kind of nuclei. So one of them sperm, one of them eggs, nuclei. So that's why it's called pronuclei, and it's a pronuclear stage zygote. So the transfers can be done at this stage. You could actually isolate this pronuclei and transfer again where? Into another pronuclear stage cytoplasm. So the donor cytoplasm has to have the same stage where you have to remove and discard this pronuclei, and then you transfer these, basically replace. So it's called pronuclear transfer. So these are kind of, I guess, 4 different nuclear transfer techniques. They all make this mitochondrial replacement technique, so MRT. So going back to GV transfer, so why would anyone do at that stage? Well, because it's a visible nucleus, so it's easy for an embryologist to see in the microscope. And also there is some other attractive moment is, you see, this is done before egg go through 2 actually divisions to become haploid. It's called meiosis. And there are some meiotic errors, as I mentioned, particularly because of cytoplasm is poor. So if we replace it at this stage, so hopefully this new cytoplasm now will do a little bit better job of controlling meiosis and may reduce meiotic errors. So that's one of the main attractions. But there are a number of disadvantages, and one of the main of them, and I will be telling you, because I'm expert in nuclear transfer, I do living in nuclear transfer. I've been doing it, for the last 30 years. So to transfer, say, nucleus from one cell to another, not only you have to have the same stage of the donor, egg, but the issue is that both patient egg and donor eggs, they're actually going through cell cycle. As you know, there's the cells go through metaphase when they chromosomes, and then they divide and become a G1 and S phase and G2. So there's a cell cycle stages. So cells actually going through the cycle, they're moving. So it's a moving target, and it's very difficult to set the same cytoplasm match, particularly with this nucleated stage, which indicates this is S phase. S phase is actually when DNA duplicates, replicates itself, and that's actually a very, very sensitive stage, which is also very long. And it's best not to do any transfers there because it can actually disturb, significantly the DNA replication. And another thing is you have to match, as I mentioned, the cell cycle stage of the donor nucleus, nucleus and then donor cytoplasm. And matching usually has to be within like 30 minutes. But the S phase is actually very long, probably 10 to 12 hours sometimes. And it's impossible to predict what where the donor cytoplasm is versus the patient nucleus. And that actually makes it impossible to match it. But if you do unmatch, which happens most of the time, you get developmental arrest because of the disturbance of DNA replication. So that's one of the main deficiency or disadvantages of these techniques doing at the pronuclear stage. I know it's easy for embryologists to see, but it will backfire because of the problem. Another thing is because of the GV is we actually have to retrieve it much earlier from patients, and that means we have to grow them or mature, we call them, into metaphase 2, into haploid egg. And that hasn't been really successfully done in humans, particularly because we have to remove the cumulus cells, which actually supports to see the nucleus. We have to discard all the cumulus cells and granulosa cells, and without them, usually maturation doesn't go well. So even if you perform this procedure, you cannot make them to grow to mature eggs. So the other issue is, when you transfer such a big nucleus, you have to actually transfer, and I'll show you how actually done with a little bit cytoplasm, which we call carryover. And sometimes this carryover amount, if you try to pick up this big nucleus, is very large. Up to 30% of the cytoplasm will be carried over, meaning you bringing this deficient cytoplasm, of course, with mtDNA with it, into the newly formed GB oocyte, which is bad. We actually want to replace, meaning you don't want to bring bad cytoplasm, remove it as much as possible, and bring maybe 1% as possible. But with a GB transfer, the same as a Pronuclear transfer, that's impossible to do, and that's why you're not really doing a replacement, because the word of MRT means replace. That means you remove bad mitochondria or bad cytoplasm completely as much as possible and bring 100% new one. In this case, I guess it would be 30% of remaining cytoplasm. Anyway, but the issue is also that safety and feasibility and effectiveness, particularly for humans, hasn't been demonstrated. I don't know really any babies were born in humans. I think the only babies were born within mice. And even in other animals, it wasn't possible to do. I, for many years, tried to do it in monkeys because I developed spindle transfer in macaques. But I started with GV and Pronuclear transfer, and those failed. And that's why, probably never progressed to humans either. So another one is, as I mentioned, it's a Pronuclear stage, which is after fertilization. It has the same disadvantages. The advantage is only one, as you could see. It's just because visible nucleus doesn't require probably special microscope. But the disadvantages are all the same. It's a moving target and impossible to match with donor cytoplasm. And also, it's done already after both meiotic divisions are completed, and that means you cannot correct meiotic errors. There are some ethical issues because the donor cytoplasm has to come from fertilized donor egg, zygote. That means you have to destroy that zygote, which is already human life. The other things is actually you have to use lots of chemicals to be able to, because these pronuclei, they're kind of tied up with microtubules to the cytoplasm. So we have to use, in this case, nacadazole, very toxic thing which completely destroys microtubules. But the problem is that it has, toxic effect. It's actually not reversible. And that's why there is a, very big chance that the embryo will arrest. And then they mentioned about carryover. Again, this carryover of maternal cytoplasm and mtDNA if it's done for sake of replacing mitochondrial DNA. Feasibility and safety and effectiveness. Again, it has been proven mostly in the mice. In humans, I don't know any reported case, even though there is seems like there's been newspapers that at least in UK, the child was born, but I haven't seen the paper. Actually, in Newcastle, they were trying to do it for more than a decade, as far as I know. And given that they haven't succeeded yet, it tells you how difficult it is. But majority of babies are born, that's proven, they actually with spindle transfer. And that's why, so spindle transfer has more advantages. One of the main advantages, as I said, spindle transfer is done at metaphase 2 arrested eggs, meaning the eggs are arrested at like 12 o'clock and they're not moving. The clock there is sitting. So is the donor egg. And we have 4 or 5 hours when we actually can transfer spindle to the metaphase 2 arrested cytoplasm, which is perfect match. And it's because eggs at that stage naturally arrested. They waiting for sperm to come in and start moving forward. And so since this is done before ICSI or IVF, so this is actually perfect time. It's a natural time to do it. And that's one of the main advantages. And that's why it actually works. So the other thing is, of course, it's a performance actually after first meiotic division. As you can see, the first polar body already segregated, but second polar body will be after fertilization. So unfortunately, we wouldn't be able to correctly if there is any ploidy already occurred in all the women's egg. But we probably can correct with a second division. So another thing is, as I said, it's very small carryover. We perfected this technique so we can carry maybe like around 1% of the cytoplasm with the nucleus, meaning replacement is about 99%. The other advantage, of course, as I said, because this has worked for me in macaques, so we defined these techniques and published many papers, and those actually today is used by IVF clinics to mimic this. There are some disadvantages. Of course, one of them is there is no visible nucleus, so you cannot just use a regular microscope, and it requires very special imaging equipment. Also requires some specific microsurgery skills, and also there is some reagents very specific, which unfortunately most of it not available commercially nowadays. We produce it in the lab mostly for research use. That thing, and the feasibility, safety, and effectiveness. So as you know, there is first time was demonstrated by Birth of Child, published in 2016. That time it was done to replace mitochondrial DNA disease, not for infertility. But in 2023, we published a paper when we showed the safety and partial effectiveness for infertility as well. Coming back to other form of nuclear transfer, which is polar body, right? So when we transfer spindle, which is somewhere over there, what will remain in a patient eggs is that second nucleus, which is just sits there. So we actually can transfer it, and of course that will be transferred into another donor cytoplasm. So we will use another. So we basically double number of eggs. So from one patient egg, we can make 2 MRT eggs, which is of course is a huge huge effect because of the number of eggs we usually retrieve from advanced maternal age patients. It could be just 1 egg, and making 2, it's a big deal. So that actually efficacy could be doubled. And if you have a 5, we can make 10. So that's one of the major, I guess, uses. So it's actually very similar to spindle transfer, but one of the things is actually it doesn't seems like has any maternal mtDNA, because at least in laboratory when we make we couldn't even find any mtDNA from the patient. And I'm going to tell a little bit about this mtDNA. Disadvantage is well, you have to use donor egg, which is in the metaphase, so you have to use spindle transfer, which is not visible. The other thing is a metaphase 2 egg polar body, which is polar body 1, is actually has a kind of short lifespan. It doesn't stay there alive for very long. So as soon as it's actually segregates, becomes separate, it's because nature usually discards this, and that's why it goes through called apoptosis, basically disappears. So it's the DNA degradation, and it has to be done very quickly after the polar body was separated. Unfortunately, most of the time we cannot control it, because this segregation of the polar body happens still in the follicle in ovary. But there is a way of stimulate and retrieve a little bit early, so we can actually try to time polar body extrusion in our incubators by watching. And that way, of course, if we done it within 1 to 2 hours after it's got separated, I think that's the best time to do. So the feasibility, safety, and effectiveness, again, hasn't been done. There are no children, as far as I know, born, at least proven and published. Even though in laboratory settings we do that and we produce viable embryos, sometimes embryonic stem cells, but that's not the babies. So that's kind of downside. This is still need to be shown, particularly for safety, because it's a little bit different than spindle transfer. So technically, how this is done by spindle transfer, I've seen that previously on this series. There has been some talks where they showed some videos, and actually, it's videos from my lab. So you have, in this case, two eggs, the same stage. They have to be metaphase 2, right? Which is no big deal because that's how we retrieve them for fertilization. They have to be fertilization ready. They usually eggs come with lots of cumulus cells, and we strip them off so we can see them. In this case, we have to visualize the spindle in a donor egg, and use kind of tiny pipette and then kind of lasers, and then we pull it off. As you could see, it's actually we pull off like a glove with a membrane, and by pulling, we actually rip off over here. So that's how enucleation is done. So in this case, it's a kind of it's artificial cell division. We don't just go and rip off the chromosomes. No, we do it by kind of dividing this egg into two. One would be small with the nucleus, another one just cytoplasm. So the enucleation is actually experimental cell division. We do the same with the donor egg. With the patient egg. In this case, of course, we retain this second egg, which we call karyoplast egg. And this is cytoplasm from a donor egg. And then we put them close, and then we induce fusion, which is also kind of, we use some natural ability of the Sendai virus to fuse membranes. And then once we do that, within 10-15 minutes, you will see that these membranes become one, and the content of this karyoplast will be flown inside of egg. So the spindle, which was in patient egg, now will become inside of the donor cytoplasm. So that's your MRT egg. And if you would do polar body, of course, it would be very similar. In this case, we take this polar body and then we transfer it the same way into another egg. So it has to be two donor eggs used per one patient egg. So as I mentioned, because it was impossible to see those chromosomes, so we actually worked with from many years ago. It was in 2005. We collaborated with a company which is called CRI, Boston-based, and they were producing these pulse scopes at that time. So we worked with them to make kind of microscope that would allow you to see image by looking just eyepieces. And this is actually picture, but the spindle imaging is a little bit different. So as I mentioned, you will go with a pipette and then remove a kind of second small cell like a polar body, and then if you spit it out, this is what you would see. See that you had one egg, and now we made it two eggs. The tiny eggs, which you could see here, that's the spindle, and another egg which has just mainly cytoplasm. So that's the technical details how we do it, and of course now we would put these karyoplasts inside of donor oocyte, which will be enucleated as well. As I mentioned, this was initially done by me before we start working with human eggs. We worked with mice and then with monkeys, and then we transferred to human eggs. But this was done with rhesus macaques, and these are one of the first macaques that we produced, and the paper was published in 2009. It's called mitochondrial gene replacement. That's how we called it, and that's how the term of MRT came. These macaques, of course, we not only these four, we produced more, but then we actually bred them, and then we showed that they not only, healthy, but they're fertile and so on. So this was very important to do like a 2-3 generation of the maintaining them, and that's how you define safety. Of course, this was done only with macaques, and next stage was for us to do the same procedure with humans, and then we did it in vitro first. But then this is a study that we published more recently, 2023, when we used the spindle transfer to basically assess mostly safety of this spindle transfer in humans. And of course, in this case, we selected patients that have problems with becoming pregnant using regular IVF. The 25 families we selected, the age was 32 to 40. So still some of them with advanced age, but not all of them. But all these families had infertility, which is proven to be resistant to IVF treatment because they went through regular IVF cycles with their own eggs. So these 25 families went through, I guess, cumulative 159 IVF cycles. Some of them went through 3 times. There were some families that went through more than 10, 11 times, as you could see. So 6.4 was, average, IVF cycles they went through. And of course, none of them were pregnant. So, and the reason were, of course, all the issues that low blastocyst rate, high aneuploidy, embryonic arrest, and so on. So they were offered to do MRT. In this case, as I mentioned, they would go through stimulation again. We would retrieve from just one cycle the eggs and do this spindle transfer. And donor eggs would come from young, fertility-proven donors that we select them very carefully. So in this case, we did the MRT just once, and so this is kind of summary of the data. Basically, many of them had a blastocyst that they never had before, so it was already good. And not only that, majority of blastocysts we tested, they were euploid. That means there was no aneuploidy that been shown before. But most importantly, after transfer, we had 7 patients become pregnant. 1 pregnancy was early lost, so there was a 6 that went to term, and 6 live births were produced. So in terms of efficacy, if you count here to 25 cycles for 6 live births, it's a 24% of live birth rate, which is probably a little bit lower, less than, for example, for young, if they would just use donor eggs or something, but still it's reasonable. But most importantly, we actually, of course, we followed up these children, and we looked at the birth rates, and then followed up for at least a few years, whether they normal, and so far that's what was reported. But we're still continuing, following up these children as I speak. So this is used for infertility, but as you know, the MRTs now also used. Actually, they were initially developed to treat genetic disease. So in this case, some moms will have a mutation in them. They inherit usually from their mom, but of course the eggs will have a mutation, so the children will have this disease. And many of them actually come already diagnosed with this mitochondrial DNA disease. At least in some countries, this diagnostics is available. So as you know, the mitochondria, it's organelles which reside in cytoplasm, and that's why when you replace cytoplasm, you replace mitochondria, and that means you replace mitochondrial DNA. So mitochondrial genome is, as I mentioned, it's another number 2 genome in our body. Even though it's very important. So it only encodes 37 genes, and there are mutations that could happen in any of these. And those are actually well-known mutations that recur all the time. And we know about 200 or 300 different mutations that children born with. None of them is actually been linked to advanced maternal age. So when I see some presenters here saying, "Oh, because of maternal age and mitochondrial mutations," I don't know any cases when all the patients had the mutations in the mitochondria causing disease in children. No. And we done some studies. We don't see any mitochondrial DNA mutations in all the patients in fertility. But these we're talking about the other category of patients. They actually don't have reproductive problems. So they're fertile, but we offer them MRT because of we can basically avoid transmission of the mutation that they carry in their cytoplasm, in their egg. So this was actually, can be used, as I mentioned, for during MRT. So here it actually shows, for example, if these patients have mtDNA mutation, usually all copies, sometimes it's half copies, would be affected. It's called heteroplasmy. But by replacing, as I said, we will take a small amount of cytoplasm with this spindle. As you know, this is a carioplast, right? So we don't rip off the chromosomes alone because they wouldn't survive. They have to be enclosed into this tiny membrane. That's the only way to transfer it. Well, anyway, so when we do that, as you know, there is a cytoplasm, and that means a few of these mtDNA molecules with mitochondria get in, which is called carryover. And when we transfer it, of course, they end up in donor, cytoplasm, and we call it carryover heteroplasmy. And we've been measuring it, how much you could avoid, by bringing too big piece, and we develop techniques. But no matter what you do, it's usually, I think, still around 1% of the mutant mtDNA will end up here, and that's called heteroplasmy. But usually 1% of mtDNA doesn't cause disease, and that's how it's been used to to replace mtDNA. But later we actually found out it's actually irrespective whether you have a 1% and 99% of the cytoplasm would carry donor mitochondria. This 1% sometimes can increase. It's actually after implantation, and this is still unknown phenomenon. We actually showed in the macaques first, and then with human embryos and embryonic stem cells, we call it a reversal. So this 1% can rise, and can rise significantly. So in this paper we published in 2023, of course, this was for infertility patients. No mutation was involved, but we still can measure patient mtDNA and donor cytoplasm mtDNA. That's actually that's what we do. So child born should have mtDNA from a donor, and that's actually bona fide of MRT. If any clinic claims that they have produced, 100 MRT babies, they have to show that mtDNA is from a donor. But as far as I know, none of them actually ever do that analysis. So that's why it's hard to prove whether these children are really MRT. But so in our papers, we actually follow up and we do genetic analysis. And yeah, these 6 babies that we produced, 5 of them had exclusively maybe 99% of donor mtDNA, which is that's why it's called 3-parent baby technique, right? But one of them actually had kind of 50-50. You see, when we measured at the early stage at the embryo, it was the 1% versus 99, but child born had like 50 to 60% of this maternal mtDNA, which is very unusual because it's probably because of a replication advantage, maybe some of the maternal mtDNA. But we don't think that it will affect in any way safety in infertility cases because this maternal mtDNA is not mutant as far as we know. But of course, if we would use it for mitochondrial DNA disease patients, that reversal is a concern. That means that mutant mtDNA would increase, and more percentage of you have in a child, so more chances of developing disease. So we actually estimate it's not very a rare event. We had the estimate when we did it before, and now we think that chances 1 in 5, 1 in 6 born child will have this reversal. Luckily, so far it hasn't been 100% reversal. It's kind of 50-60, but still that's a concern, and that's why this is a cautious, I guess, efficacy issue for those patients seeking with mtDNA disease, seeking to do this MRT for prevent transmission. And so the other, I think thing is, as I mentioned, the MRT was actually first used in 2017 to 2016 to do to prevent transmission of mtDNA disease. In this case, the mutation was at this position of mtDNA, which is well known mutation causing very severe disease called Lee syndrome. And this case, when they measured it at the blastocyst stage, they still had like close to 6% of mutation load. Well, this tells you how experienced the embryologists were. Usually it should be below 1%. So having almost 6%, meaning they were carrying too much of the cytoplasm. So that's how they end up. But born child, at least, when they measured in foreskin, had actually a little bit increased, kind of already 9% or 10%. So unfortunately, there is no any follow-up. This child, whether it's really healthy, whether it has developed any syndrome, that's unfortunate, because most IVF clinics, after they transfer embryo, the job is done. But with MRT, because it's still experimental, we have to follow up, particularly with children, and do genetic analysis on this reversal and so on. So it's still not sure if this baby is born, but at least it's been reported, and it's considered to be one of the first cases of use of spindle transfer for prevention of mtDNA disease. So when we do that, of course, the other questions, not only the amount of residual, what we call carryover, but also, whether it will stay the same, like 1%, and how to prevent it to become 60% or 100%. So that's still work in progress. We're trying to figure it out. But the other thing is, as always, comes as a question, whether it's for mtDNA moms or it's for patients with infertility, is whether donor mtDNA that will be replaced in the egg, and that means the child will have a mtDNA from a donor, has any effect on a child. Well, particularly because there is a sequence difference, right? It's mtDNA is a DNA, and then by sequencing, we know what letters are different, and that's what we usually look at, whether we have to match or not. Matching, that means you have to find donor egg donor that would carry exact the same mtDNA, exact the same haplotype, which is a little bit difficult to do. But if you do kind of mismatch with any given donor, whether it's going to have a problem or not, we actually addressed it in monkeys. I mentioned to you about these macaques. The first macaques was Mito and Drecker, and then so on. Actually, they were done with donor mtDNA from actually different subpopulation of macaques. So we call them Indian Chinese, and they very different in terms of single nucleotide polymorphic sites that from donor and the maternal. So say like more than 700, which is actually huge when we look at the humans. Actually, humans, if you just do random, I think they may not differ more than 20 to 40 SNPs. So I think the maximum possible distance between some human mtDNA subpopulation could be maybe 100 SNPs. Of course, I never seen myself, but even if you do that, so you could see that in the macaques we had like completely different mtDNA haplotypes with so much difference, and they still didn't have any problems. They grew to adulthood normally. They had babies. So in my view, I think any human mtDNA haplotype, haplogroup would work. It shouldn't matter, how different or distant it is, as long as it's, of course, human. You don't want to use some other species. So that's in terms of whether we have to match donor mtDNA haplotype or not. But of course, we have to look at the quality of the donor egg and its ability to produce viable embryo and pregnancy and baby. And that's actually one of the important things when we select donor eggs for MRT. So I think I'm going to wrap it up, my presentation. So because already had so many things covered, but I'm hoping that we'll cover more uh, Q&A. And so I would like actually to thank many members of my lab. Many of them actually already left my lab and have their own labs. So one of them is Don Wolf. He's actually my mentor. When I came in 1998 to his lab, he had the Primate lab, and I started there. And then from then I had so many different people that, particularly Masahita, he was a key in developing this spindle transfer technique, and many others. So with that, I think I'll wrap it up, and I'll try to answer any questions that you might have.
Brilliant. Thank you so much. That was very, very interesting. Lots of details. And thank you so much for walking us through all aspects, but also through all those various options available, how it works. So thanks a lot for that. And if you see it, there are plenty of questions for you in the chat. So let's get to them. I know that you are definitely waiting for the answers. We have around 15 minutes, 20 minutes, so we will not probably able to answer them all, but let me go ahead with the first that we have received. If you can take a look at this one. I have read your research of the MST embryo's strong potential for self-correction. Our embryos after MST came back with full chromosomal abnormalities, but it is perfect day 5 for AA embryo. Do you think we should transfer it? Anything you can?
I think in the beginning of my presentation, I already told you about this PGTA, which is not reliable in terms of detecting aneuploidy, just because it's a very tiny amount of sample. It has to go through so many processing. By that, usually we lose chromosomes, during amplification. So that's one of the reasons that PGTA is not reliable. And plus, it's actually because you rip off, 20% of cells from the tiny embryo, so it's not safe either. So in that case, unfortunately, I cannot comment. But of course, if you already did that analysis, and if I would still, see maybe to do repeated analysis in different place. But for MRT, for that main reason, we actually don't do any PGTA, but we transfer the, of course, viable blasts, but we request patients to go through prenatal testing. So prenatal testing is more reliable because there are more cells there, and we get, 100,000 cells in biopsy. And there are 3 different stages when you could do. Some of them actually you could do through blood of the mother. It's a kind of non-invasive, and then you could do, if that doesn't come, you could do CVS, and then if that still comes was questionable, you could do amniotic fluid. So those are more predictable ways, and then you could decide.
Okay, thank you so much. There were some questions on PGTA here. So thank you for explaining, because there were questions regards to this. Someone even mentioned that some clinics offering MRT insist on PGTA testing in order to go forward, for example, right? So this is the comment we received as well. So thanks a lot for adding this. And now we will go for the next question. Okay, there are questions, very similar ones regarding the age. In your studies, you say, I think that there was 32 up to 40.
Yes.
Is there a particular reason? Someone asked about 45, for example. There is also someone who is 40. Are you able to summarize and tell us how it
Yes.
works?
So at that particular study was done under 40. This was a kind of decision on regulatory agencies to not to go past 40, but we still planning to do more advanced age. But yeah, so far data is under 40.
Okay, so for now,
and
at least.
that efficacy 24%, I guess, applies to that age group. And if you are older, so it's still unknown, how effective would be the spindle transfer for older patients.
Okay, I think that's clear. Thank you so much. There were actually a few questions on that, so we have it
Yeah, I know
covered.
that's actually very frequently patient ask about what I should expect, what would be efficacy. Well, unfortunately, it hasn't been done a lot, so that's why it's at this point it's unclear.
Okay, and let's have a look about on this question. Sorry, when you use donor cytoplasm, there might be the chances of transferring their small genetic cargo content to the patient. What is your take on this?
Yeah, so microRNAs and then other RNAs, they're actually part of this, what we call is maternal factors, which is could be totipotency factors. So they must come. That's why we remove them from a patient egg, but without it, the egg wouldn't develop. So they have to come with donor egg, and it's a part of the cargo. Whether you want it or not, it will be there.
Okay. Thank you so much then for that. And more questions are coming. So thanks a lot for that. I'm trying to look through all of them and trying to select the most. There were actually a few that were regarding your presentation, actually. So let me just go to that one. It was really interesting here. Yeah, if you can explain how this maybe, what does it mean 50% euploid blastocyst, right? 50 of 100 blastocysts, what is the rate for one? This is something we discussed.
Yeah, I think I have to take a look, but this was a kind of when we estimated aneuploidy. So it was, I guess, 50% of blasts we tested they were euploid, and that's why they were transferred. So which is actually probably expected rate, and otherwise, I think most of these patients when they did the PGTA before, 100% the blasts, if they actually reached to blasts, were unemployed. But as I mentioned, any PGTA testing could be erroneous because of the technical issues, and cannot really confirm it. And that's why, in my view, I think it's best not to do it, particularly given that MRT blasts, they're kind of weaker, they're smaller, and plus they already have so many holes. And trying to do biopsy, usually you rip off the whole blasts out of the zona pellucida, and that happened a lot. So that's why I think it would be safer not to do biopsies at the blastocyst stage and do it prenatal, as I mentioned.
Okay, thank you. Again, crystal clear, this one. Do you have more insights on mtDNA reversal and possible mechanisms involved?
Um, yeah, I think I published some review papers on that, particularly saying this is probably one of the basic biology of mtDNA. With nuclear DNA, we know every chromosome, every gene will be replicated only once, and that's how daughter cells will be genetic copies of the mother cell. That's why it's called cloning. That's why. But with mitochondrial DNA, unfortunately, there are could be an egg, could be 500,000 copies. But that doesn't mean that every of these mtDNA will be replicated only once, and then divided and equally distributed to daughter cells. So apparently, in mtDNA cells, I don't know how nucleus or whatever decides that only 1 or 2% of mtDNA will be used for replication, but they will replicate many, many thousand times. But the rest of them, that means 99% of mtDNA, will not be used for replication. They probably will be used for, transcription, translation. And that's how you could explain that how come this 1% all suddenly will become 100. Maybe those mtDNA that close to the spindle, the nucleus, they actually selected to be a replicative mtDNA. We call them germline. But it's still kind of I guess, theories to explain why we see this phenomenon. mtDNA biology is quite different than nuclear genome, which is Mendelian. That's what we're used to. And in this case, I guess it's a different genome, completely different laws it goes with. And unfortunately, we know very little about it. So here it comes. Actually, people who were studying mtDNA biology didn't know about it until we started doing treatment. And that's how we found out. Well, they couldn't know it because only by transferring and different haplotype, you would know which one actually ends up in the baby.
Okay, thank you so much again. There are plenty more questions coming up. Many questions are quite similar on this, right? So maybe you can tell us what is this process with this process not being allowed in the US? Where can we travel? Do you work closely with any clinics outside that you can recommend? There were questions about Mexico as well for this.
Mhm.
So anything
Yes.
you
So
can advise.
unfortunately, yes, in US, it wasn't really banned, but the FDA was banned from reviewing and allowing clinical trials. So that's how we cannot do it. So it's in many countries. So the other countries where it's kind of officially allowed, it was UK and I heard Australia, but they only allow it for mtDNA patients, not for infertility. They still kind of waiting for the data we produce to show them that it will be effective for infertility. But we have to continue doing clinical trials, and that's how I've been involved with the clinics in Europe and wherever they allow them to do, or they don't ban, but we do it like a more clinical testing, a pilot study. So one of them was in Greece, and I see some of the questions saying, "Do you have a plan?" Yes, we have plan. We still want to do for older patients and maybe do larger trial. Still kind of in the plans because we're still waiting
Mhm.
for approval from the regulatory agency there. But there are some other clinics that seems like they wanted to introduce it, but we in agreement saying let's just do small pilot study and see if it works, and then I guess you could introduce it as a general or regular clinical treatment. So I'm particularly working with a clinic in Cyprus, in northern Cyprus. That's where we do small kind of pilot study, where we actually, myself including, perform it because, as I said, it requires lots of skills. So we want to make sure that we do it as we developed it here.
Mhm.
So there's also a clinic in Kazakhstan, Almaty. They actually want to introduce in that country, and I will also want to do some small clinical study there. So the Mexico, I think they also wanted to do, but not sure if if they get all the permits or whatever is. But if they also want to do this, would be good because I know there would be lots of patients from US and Canada go there. So those are, I guess, only three clinics where I officially work with. We do ourselves, but it's a small clinical study and done for kind of uh, um, with limited number of patients. So just we can publish. That the question is, what about 45 years? What about 50 years? Hopefully, those small, 20, 30 cases we do for these particular groups, and we can say, oh yeah, we have the same pregnancy rate or white birth rate, or maybe lower. Still kind of waiting
Yeah,
for those
exactly.
results. Yeah,
Right.
lots of moving parts to organize these pilot studies.
Okay. Thank you so much. Brilliant. Because as you saw, there are lots of questions. I
Yes,
will
yes,
ask
yes.
you this as well. This is also interesting. Can you use our skin cells to make an egg and use that egg as our own donor? So transfer our nucleus to that healthy cytoplasm from skin cells, created egg from the stem cell. And that was the question is also about Mexico or Cyprus,
Oh,
right?
okay. Yeah. So using skin cells to make this egg, it's actually a different technology. It's called IVG, in vitro gametogenesis. It's just making from any DNA from your body eggs. Since if you don't have your eggs, and many patients by 50 or later, they will have a completely lost the ovarian reserve. So that technology, unfortunately, is still in development. Yes, we're working on it. We already produced mice, but it's still early to testing with humans. We're still kind of doing in vitro. We can do here in my lab with human eggs and embryos, but without implanting it. And that
Mhm.
work is done. We have a paper submitted for publication. But I don't think we will be ready anytime soon to do clinical testings like it was MRT. MRT I developed in 2005. We published paper in 2009, and you could see how many years passed. It still kind of cannot finish these clinical trials, right? So with this IVG, it's going to probably take much, much longer, and probably
Mhm.
regulations are going to be much, much tougher than for MRT. So that's bottom line. But I know that would solve lots of problems, because right now
Sure.
I think when we're trying to test MRT for advanced maternal age, the major issue is number of eggs we're getting from them. It's sometimes one or none. So how you can get 25% live births if you have only one egg? When we did it in Greece, I don't recall, but average number of eggs probably was there around 5, 6. So that plays a role.
Okay. Thank you so much indeed for that. And there were some questions again about Cyprus, and if someone would like to join the studies for older patients, how does it work? Are they able to get in touch with you, perhaps? How does it work?
Yeah, I think they could do probably in touch with me, and we will usually review the past history, infertility, and then I would refer them to one of these clinics where they would go through stimulations, and we do it just like we did in Greece. They go through stimulation and they freeze their eggs. And then usually we would plan to do MRT later. So usually I travel with my team there. And of course, the local clinic will start preparing fresh donor eggs. And we
Mhm.
select those also very carefully. And then we style donor the patient eggs, and donor's eggs are fresh, and then we do perform MRT. That's how it works. But
Perfect.
they have to, of course,
Mhm.
fit some of the
Yes,
inclusion criteria. Yeah,
that's
most
always
of
the
them is
case.
they have to already, unfortunately, go through lots of regular
Mhm.
IVF, because we have to have a proven record that IVF regular doesn't work for them.
Yeah, okay. Thank you so much indeed for that. I will share Professor Mitalipov email address in the chat, so you will have a chance to also get in touch directly. We will be slowly finishing. I think that we can have a look at a few questions more, and we will be finishing. But thank you so much for those so many questions. The question is here: Can you talk about what is unknown about the impact of these procedures on born children?
Yeah. So as I said, we only still following up on these 6 children that were born in Greece. So in the paper, I think they were about 3 to 5 years when we published the paper. So we had like at the early, uh, you know, past prenatal, and then whatever the checkups they went through. That's part, but we're still following up. And of course, at this point, we don't see any adverse effects. Everything seems normal. They go through normal, but that's only way. Usually, these type of studies are long term, because yes, of course, patients are going to say, "Well, this is early. What about, are they going to develop anything when they adults?" So we have to wait until then. Are they going to be fertile? Well, we have to wait for that too. We had to wait for, I don't know, 40 years until Louise Brown had her baby. So I guess lots of these questions, unfortunately, cannot be answered right away. So we're trying to do it. Of course, we did it in macaques. We did 2, 3 generation, and we published that paper. So that one we can use, and that's why was important for us to move on to humans. But the other thing is, I know there are lots of other clinics. They claim they're doing MRT, but of course, whether they're doing it or not, I don't know, because it requires very specific skills, equipment, reagents, which they don't seem like have.
Mhm.
But they still advertise it. Most importantly, even they claim they have a babies born, 40, 50, some of the clinics, but they have to be following up them, right, and telling them what happened to them. But that's the issue with the clinics, because
Mhm.
we don't want to, this to become regular procedure until we do testing. And that's why these clinics that we're doing this pilot study, the patients has to agree that we have to collect data on unborn children. Of course, most of the tests are non-invasive, regular checkups, maybe occasionally blood, but that's that's how information on safety would be gathered, and it's still work in progress.
Okay, thank you so much. So that's why it's very important to remember you have to be cautious as well,
Right.
as Professor Mitalipov said, right? So even though you see that they are advertising MRT, might not be actually the case. So we definitely should be cautious when it comes to that.
Yeah, that's unfortunate that the misinformation and patients travel there, and maybe they do some procedures. Sometimes it's not really MRT, but that's
Yeah. All right, perfect. Thank you so much. Probably our final question for today, I will display it for you. In the 2023 study, was that 24% outcome after transferring every good embryo developed, or was it the first transfer and they got other attempts with subsequent transfers of other euploids made in the initial MRT procedure, and it isn't captured in the 24%?
I don't think there was well, there's probably some blasts left. I don't recall now that have been transferred. They could be to the patients that already had pregnancy with prior transfer. There might be some still remaining embryos. I don't remember that, but yeah, I think we did. We had to wrap up the study within 2-3 years, and at that time, this is what the number of embryos we transferred and pregnancy had, and babies were born.
Okay, thank you. Thank you so much indeed. And as I've mentioned, that was our final question. I see there are more questions here, but as I mentioned, if you have any other questions, you can get in touch with Professor Mitalipov directly, and I'm sure he will be able to provide you with more details. That was a great session. Thank you so much for your patience and for your time today. I do hope you also found it interesting for you with all the questions here.
Absolutely, yeah.
Any final words before we finish for today?
Well, I think as you mentioned, there would be lots of patients actually listening, and that's what I try to direct my presentation towards them to explain them, and I'm glad that hopefully at least some information was cleared out. But of course, there are so many more questions, and that's why we have to continue doing clinical testings, trials. And patients, at least who's signing up, they have to agree that they have to provide with data on their babies, because sometimes they just don't want to do it, which is I understand. But this is important for upcoming patients, for future patients, that we do these studies, and we actually follow up lots of children, particularly with mitochondrial disease. So it's very important for everybody, and I really appreciate most of the patients that so far sign up for these trials are actually very supportive of the science, and
For sure. So we can explore it more that way as well, of course, right? Thank you so much indeed. And as Professor Mitalipov said, you can get in touch, and if you have more questions, and I'm sure you do, if something you wish to discuss, you can go ahead and do it. Thank you so much for today. Have a lovely evening, everyone, and Professor Mitalipov, have a great day as well. And of course, we will be back with more events. So again, it was great to have you. Take care and good night. Bye.
Good night. Bye. Thank you.